So this is the 8th lecture in a series of 8. The topic for today is trial closeout and reporting. In the previous 7 lectures, we've talked about sort of why we do clinical trials, defined the questions, considered some basic trial designs and how you size the study, reviewed some of the requirements for trial monitoring for safety and benefit. And in the last lecture, we spent a great deal of time looking at issues in the data analysis, not methodologies, but issues that you need to take into consideration, such as analyzing all patients randomized, all events that occur, being careful about subgroups, and so forth. In today's lecture, we're going to focus on the issues of closing out the trial and reporting those results in the literature. So we're going to first cover the issue of patient and trial closeout. Both of these topics are important because it's tempting when one gets to the end of a trial to rush to the publication, to the presentation, and sort of forget about some of the details that need to be taken care of in the final stages of the trial. In the patient closeout, one of the things that's really important is to have a final patient visit where you complete and tidy up all the outcome assessments, collect any missing data that might exist. You certainly want to keep track of vital status and key events, safety events that might have occurred. It's also an opportunity to transition the patient from the trial to the post-trial era and how the patients will be treated and taken care of. Also, it's a good time to notify the patient how they will find out about the final trial results. The ICH E9 glossary, that is the International Conference on Harmonization document, talks about the intention to treat principle with regard to patient closeout. It has the consequences that subjects allocated to treatment should be followed up, assessed, and analyzed as members of their group irrespective of their compliance with the planned course of treatment. We talked a great deal about this in the last lecture, but it's important here in the final patient visit to ascertain key elements of vital status, key safety, serious adverse events, and so forth. I want to share with you just one example to show what not having this taken care of can cost a trial. This is a trial called the companion trial. It's a trial evaluating a pacemaker or defibrillator compared to best medical care in a population of chronic heart failure patients. This paper was published in the medical literature. This is the summary of the study design of the companion trial. Patients were enrolled, tested at baseline, and randomized to one of 3 arms: the pharmacologic therapy alone, the pacemaker plus best medical care, or pacemaker and defibrillator plus best medical care. The primary endpoint for this trial was the composite of mortality and hospitalization for heart failure. And the definition of heart failure is given here in some detail, requiring a certain length of inotropic or vasoactive drug. The second endpoint, at least the highest ranking second endpoint, was all-cause mortality. It had a statistical plan with intention to treat. Endpoint collection begins with randomization. It was an open trial, obviously, because of the nature of the device. There was a steering committee, endpoint committee, many good design features. There was 2 comparisons here: the pacemaker versus best medical care, or the combined pacemaker defibrillator compared to best medical care, and the alpha level, or the type 1 error, was allocated between the 2 in an unequal fashion, an option which one can do if one choose. And the sample size was based on a 25% reduction in the primary endpoint and a 25% reduction in mortality. The first patient was enrolled in January of the year 2000. And in November of 2002, the monitoring committee recommended to the steering committee to stop enrollment because the number of primary endpoints had been reached, or was at least projected to be reached by the end of that month. And in fact, some efficacy boundaries had been crossed. So the steering committee, in fact, stopped enrollment on the 18th of November, and with the follow-up to be concluded on the 1st of December of 2002. However, it was already known that there was a problem. That is, there was a disproportionate withdrawal rate and therefore a disproportionate follow-up rate between the arms. The best medical care arm had a 13% withdrawal rate compared to a 2% in the other 2 arms. Because of the nature of the way it was withdrawn consent, it meant that follow-up data on those patients was not available. After much deliberation, the steering committee and the data monitoring committee both agreed that we needed to reconsent the withdrawn patients to collect endpoint data, because it would be very difficult to defend a study where you had 13% missing data on one arm, the control arm, and try to have any valid comparisons with the defibrillator or defibrillator plus pacemaker arm. In 2003, in the fall of 2003, the final data were presented to the Heart Failure Society of America meeting, and the process of collecting the endpoint data and vital status was as complete as going to get, but yet not perfect. The best medical care arm still had only 95% complete ascertainment, whereas the other two arms had 99%. It took 6 months to recover all of the missing data. Just for completeness, I'll show you here the final results. This is the secondary endpoint of mortality, and as you can see, the two device arms both are superior to the best medical care arm, with the defibrillator pacemaker arm being the best overall. With regard to the primary endpoint, which is a combination of mortality and hospitalization, you can see that the two device arms look very similar, but both of them are superior to the best medical therapy alone. So these trials were successful in showing that the devices had an improvement over the best medical care. But the lesson for this lecture is that the process of having incomplete data at the close of the study required a reconsenting of those withdrawn patients, took a long time, several months in delays to getting the results complete and presented. It would have been better had we had a plan in place to address the withdrawal rates in the first place, and then once withdrawn to reconsent for at least very minimal follow-up of survival status and hospitalization. The conclusions of the study, of course, was that the two devices reduced mortality and hospitalization, and the device defibrillator arm reduced mortality. So in that sense, it was a very successful study, but nevertheless, a lesson was learned about closeout. Now, in the trial closeout, we need to have plans for transitions for the patient care, your clinical staff, and the central units, laboratory units that you relied on, and also some plan for data archiving. Very tempting, as I said earlier, to come to the conclusion of the trial with all the excitement about reporting results and not plan for the transition of the trial and archiving data. Why would you want to bother archiving data? Well, first of all, you may have some opportunity and need to verify the results. There may be a reason for a post-study follow-up, long-term safety, and trials of these kinds often serve as a good data source for just general research on methodology or generating new questions for the next study. In thinking about keeping adequate records for trials, I've listed here several items that need to be taken into consideration. You want to keep a protocol. You want to have the manual of operations if you developed one, and by that I mean the document that tells you how to make some of the measurements, how to do some of the adjudication, with the rules of the trial. You need to have a clean set of the data forms so you know exactly what data you collected. And in some way, a copy of the completed data forms. It might be an electronic version. Almost certainly today that would be the most common. A copy of the final edited data file, documentation of the analysis that you conducted, data reports and the minutes of the data monitoring committee meetings, if there was a formal data monitoring committee, and key steering committee minutes that would document why you made certain decisions and why you defined things as you did. This is just a suggestion, but you need to be able to have enough documentation that you could go back to the trial several months or perhaps years later and reload and begin to understand what it was you did and why you did it. Sometimes we go back to a trial to look for the long-term follow-up of the patients, assuming that there's funding available to do that. You may want to monitor, for example, how long did the benefit last, or to look for long-term late benefits if a trial did not show an early benefit. And finally, to look for long-term toxicity that was not evident in the shorter-term trial. Whatever the reasons, when one goes to follow patients after the trial has been formally closed, one will depend on the data and the archival information that I just described previously. And then, of course, there's the use of the data to generate new research. You may identify new risk factors. In today's world, you may have some biospecimen data and begin to examine the impact of genomics on your research. You may want to design new trials, or you may want to use the data to check out and test a new methodology, perhaps a statistical method for analyzing the data. So this is a valuable resource that trials can provide, assuming that the data is put away in an orderly fashion. Now let's talk a little bit about the reporting of the trial. As I indicated earlier, we can design, conduct, and analyze a trial in the best possible way, but still we must report it in a way that would be justified to the trial that we just did. I'll talk a little about the rationale, some of the guidelines, and comments on those guidelines. Reporting trials, of course, are important. It's a way of sharing the information with practicing physicians because they rely on the literature to keep current. And accurate reporting is important in deciding whether you adopt a new therapy or modify existing therapies that are in use. Because of the importance of reporting trials, there were several proposals for what would be required in the reporting of such trials. This was discussed in the literature, including the references I've given here. A JAMA editorial in 1995 suggested that the various parties that had proposed requirements produce a unified statement. And that became known as the Consolidated Standards of Reporting, or CONSORT, which was published in the Journal of the American Medical Association in 1996. I won't go into great detail, but just share with you some of the highlights. The intent of the CONSORT document is to make it clear the experimental process, whether there are flaws or not, so that users of the data can make a better judgment about the validity of the data that's presented. And so in the website, there is some checklists and figures that are suggested to be included in the reporting of a trial. This has been widely adopted by medical journals and, in fact, is required by many of the journals and available in many languages. So we have in our current setting several requirements for reporting. There is the regulatory setting, and the International Conference on Harmonization has guidelines about reporting. The academic journals have the CONSORT documentations, and these guidelines should be taken, I think, seriously not only in the reporting of the trial, but also think about them at the design stage. Much of what we've talked about over the past 7 lectures are consistent with the regulatory and CONSORT guidelines. So here are some of the common elements that are included in the CONSORT document to describe the population under study, to present enough details of the therapy so you know exactly what it was that was being tested, to summarize the experimental design, to define or present the primary and secondary outcome variables, to have a discussion of patient accounting, a description of the quality control procedures that were used to get high-quality studies, the statistical analysis that was conducted on the data, and then depending on whether this is a randomized or non-randomized study, to provide enough details so one understands that aspect. Interestingly enough, one requirement is not discussed, and Kurt Meinert, who is one of the early pioneers of the modern clinical trial, randomized clinical trial, remarks about this in a JAMA article in 1998. Dr. Meinert states that clearly the single most important count in any trial is that the person's randomized, which serves as the denominator for analysis by treatment assignment. Surprisingly, this number is not among the counts requested. That is, they do not discuss the need for the intent to treat principle. He goes on to comment that withdrawn or withdrawal in the context of trials is best reserved to use as a technical term in relation to analysis. Its use regarding treating or following up a person in a trial is unfortunate. Withdrawn with respect to what? So he's raising the issue here that we've talked about in the last lecture, that if you do not have complete follow-up, one can, at the very end of your trial, allow bias to slip in, and we know that bias in this context can be quite powerful. So his recommendation is that in a primary analysis, in any report in a trial, should include all persons, all events that are related to the follow-up and not withdrawn from analysis for any reason. Now, in reporting the trials, we need to cover certain basic areas to describe the overall plan, to report the results. All trials have problems, and we should confess to them instead of trying to hide them. It's better to tell us what went right and what didn't go right. And finally, one has to be very careful to report the data results objectively and not put any spin or hype on our results. It only detracts from the scientific validity and quality of the trial when we do that. So here's just some issues that I think need to be covered. First of all, we should identify who did the trial, identify the clinical investigators and the institutions so we understand something about their experience and reputation. We also need to identify the sponsor, whether it was a federally sponsored trial or an industry-sponsored trial, how were the data managed, and who did the analysis. In today's world, these are critical components as one reads an article reporting the results of a trial. As we said at the beginning, you need to understand the background for why this therapy is being evaluated in the first place. Is there some relevant biochemical theory? Is there animal work that justifies why this might work? What are the results in the phase 1 and phase 2 trials? Are there other large studies in this general area that we need to know about? Or are there other pharmaceutical analogs? In other words, give me some setting in which this trial is taking place and why the trial is being justified. We need to have a method section which defines the details of the protocol in a very concise fashion. What were the outcome variables? What are the criteria for patients to be entered? What were the inclusion and exclusion criteria? If it's a randomized trial, what were those randomization procedures? What's the sample size justification? This can be a very short paragraph, but concisely written can be very important. Sometimes trials do not report the reasons for the sample size used. Of course, one needs to define in detail the treatment and the control that are being used in the trial. Moving on to other issues, we need to define the outcome assessment and blinding if it was appropriate. Measures of patient safety and adverse event. Were there predefined subgroups? And were they ranked and limited? We talked about in the current setting that we must have a monitoring plan for our trial. Some trials, phase 2 trials, may in fact require a data monitoring committee, a very elaborate plan. And finally, we need to define ahead of time what the analysis plan was, at least in a summarized fashion. What was the primary statistic and the primary outcome, for example? We need to define what the primary question was. We talked in the analysis section how a previous trial changed the definition of their primary outcome during the trial from total mortality to sudden death. We need to understand that the question was defined in advance and not changed. We need to define what the response variables for that question are. We discussed in some detail that one can have a general question about, say, the size of an infarct, but how do you measure the size of the infarct? Or were surrogate outcomes used in place of the clinically relevant outcome? We need to define what methods we've used to minimize bias, such as masking or blinding of the treatment assignment, or at least masking or blinding of the evaluation of the patient, whether it be some sort of an X-ray, an EKG, or some other laboratory assessment. We need to define the treatment groups, whether the experiment or treatment that was being tested versus the control. Was there a dose escalation? What happened when patients experienced a toxicity? Were they withdrawn from the trial? Were they withdrawn from treatment? Were they withdrawn temporarily and then restarted? How was this process handled? Of course, we must describe the results of our trial carefully. There needs to be a full accounting of all patients entered on the trial, the completeness of follow-up, what was the loss to follow-up, LTFU, and what were the withdrawal rates from the treatment and perhaps from the trial. We need to have a comparison of the treatment groups as assigned on their baseline characteristics. There's two important pieces of information we learned from this. One, what kind of patients did we study? And two, were they comparable? And finally, we need to describe our results in simple comparisons that are understandable to the readership of your paper on the primary outcome variables, using, for example, means, proportions, graphs, some measure of statistical precision, and p-values or some other measure to assess the significance of those results. We also need to have some description of how data were handled that were missing or were lost to follow-up, or how was the non-adherence handled. There should be a serious discussion about allowance for multiplicity of the number of interim analysis, the number of endpoints, perhaps a number of subgroups, and finally a thorough analysis of the side effects of the data. Among the things that we need to look for are consistency of results among investigators and centers, not in a statistical sense, but in sort of a general sense. Are there other independent studies of the same drug or analogs that are known and are these results consistent? Are there subgroup consistencies? And are there consistencies among the number of primary and secondary outcomes? Finally, in the conclusion, we need to have a brief summary of what we believe the results to be. We should summarize or generalize the strengths and weaknesses of our data. We should also comment about how generalized ability of our data. And finally, what are the trade-offs and the side effects, the risk-benefit ratio, and come to conclusion as to where this therapy fits into the practice of medicine. So over the past, 8 lectures, we've been covering many issues that are important in the design and conduct and analysis and now in the reporting of a trial. If these basic principles are kept in mind, I believe that one has a very good chance of having a trial designed, conducted, analyzed, and published that you will be proud of. Many of these issues are interdependent, but nevertheless, I've tried to break them down into simple steps that one can keep in mind in this process. I wish you well in your career as clinical trialists and look forward to seeing your names and results in the literature. Thank you very much for the opportunity to share these principles with you. So I want to thank you for the opportunity to share these principles with you. We've covered many details. I believe that if you keep these in mind, you will have a good chance of a successful trial in the design and conduct and reporting. Obviously, these issues overlap, but if you keep them in mind, I believe it'll be quite helpful. I want to thank you for sharing the time with me to discuss these issues. I wish you well in your career as clinical trialist and look forward to seeing your trials in the literature. Thank you very much.